The purpose of our experiment was to examine the food chains and webs in the ecosystem,
to collect data over the series of weeks to analyze the condition of water, soil, and the plants,
to look at the producers, consumers, and the decomposers,
to identify the hydrologic nitrogen and phosphorus cycles,
and to look at the biotic and abiotic factors.
Our Ecocolumn diagram. Here we have our plants that grew. Plant 5, plant 2, plant 4, and plant 1 all grew, but plant 3 died. I've labeled our worms, our soil, our roots. We have our filter, which was sand, and then we have our aquatic area, which had our betta fish, our gravel or rocks at the bottom, and then our aquatic plants. And our biotic factors are plants, worms, betta fish, aquatic plants, and roots. And our abiotic factors were the soil, the sand, the water, and the gravel at the bottom.
For the plant growth graph, only 4 of the plants grew. 2 of them grew pretty tall. But around week 3, most of them dropped off. I'm guessing we missed a water day or something and they wilted. So the plant growth really dipped in week 3. Yeah, that's just the plant growth graph. Here we go. All right. For the water, nitrates, and nitrites graph, well, for the first 4 weeks, we didn't see any change in any of the nitrates or nitrites besides week 1, where the nitrates went up to 0.1 for just the week 1 and then back down to 0. And then in week 5, the nitrates jumped up to 0.6 while the nitrites stayed at 0. This is the graph. For the water dissolved oxygen graph, this one gets a little crazy here. So, week 1 starts off at a good 4.5. Then 2, it dips down to like 3.5. Then week 3, it jumps all the way up to 10. That's probably when we added our fish, or the plant and the fish. That's exactly what happened. That's why that happened. And then after that, it dipped. It was slowly declining through weeks 4 and 5. But yeah, that's basically it. There's the graph. Perfect. Cool. All right. The soil fertility graph. Now this one scares me a little bit here. Week 1 through 4, great soil fertility at 3 all the way across. Week 5 dropped down to 1. What are you plucking them out? Okay. So in week 5 it dropped all the way down to 1. I'm guessing that this is from our plants dying or our worms dying, so the soil fertility went down. This is the graph. Now the temperature graph, not really much going on here. The soil temperature stayed at 24, between 23 and 24 the entire time. Degrees Celsius, that is. Now the water temp fluctuated a little bit between 22 up to 24 in week 3, down back to 22, and then back up to 24 in weeks 4 and 5. This is probably due to changes in temperature of the classroom and adding our fish and plants into the water. Here's the graph. The water pH graph. This one looks good because everyone knows 7 is the perfect pH. And that's what water should be at. So weeks 1 through 3, water was right at 7, right at it, which is great. Week 4, on the other hand, dropped down to 6, probably because in the week 3, we added our fish and plants. That probably changed the acidity of the water, if you ask me. And then it stayed like that for weeks 4 and 5. Here's the graph. Our last graph of the day. I'm sorry, this is the last one. Your MacBook Pro turned off. Soil pH. Again, great pH, 7, right where it should be. And it stayed at 7 all 5 weeks. No big changes in the graph. Here it is. That's all for me. Thank you. Hello again, it's me. So, for our adjustments that we made to our Ecocolumn, one big thing we noticed in that Miss Jenna Brunette told us about was we were watering our plants too much. We were drowning our worms, we were drowning our plants. So, we started watering less and less, and guess what? Our plants grew and flourished with this less water. They flourished, grew over 35 centimeters tall, tallest in the classroom. Tell us that ain't great. Tell me that ain't great. That's great. All right, now, that was our big adjustment. Observations. Now, after week 3, our plant started to wilt a little bit. We did move the plant around that time to a different light source when they started wilting. I think that played a big role in it. Also, they started wilting about the same time that our soil fertility went down. As you can see in this graph, soil fertility took a big drop around week 3, week 4. Now that, I think, caused our plants to receive less nutrients and start to wilt a little bit. Also, it shows up in the plant height graph where they start dropping in height because they're wilting. The 3 different habitats, the terrestrial chamber, the filter chamber, and the aquatic chamber, they affected each other so when the plants died or the worms died and created nutrients, they were being passed down through the water to the filtering chamber and then passed down to the aquatic chamber where the fish, our little beta fish Scooter, would have nutrients in the water so he could survive. And the aquatic plant, it would help them survive as well. And yeah, that's how they all affected each other. Great to see you again. This is probably my last part of the video. I'll see y'all soon.
These are the analysis questions 1 through 4. Question 1 asks, propose 3 possible reasons why there are such large differences between the ecosystem columns in the lab. Reason 1 would be access to light. Multiple people had different types of bulbs and different angles of sunlight was hitting their plants or their ecocolumn. And halfway through ours, we switched it to a different area with a different light. Reason 2 would be different types of plants. Each group got to choose which plants they wanted to plant into their ecocolumn, which could be absorbing different types of nutrients for other plants that wouldn't be able to absorb them. Reason 3 would be different types of soil. Each group had to choose their own soil, so they brought in their own, and nobody had the same. Question 2 asks, identify and briefly discuss the biochemical cycles which are taking place in your ecocolumn. The 4 cycles in our ecocolumn were water, carbon, nitrogen, and phosphorus. The water cycle occurred when water we poured into the terrestrial chamber was absorbed by plants and the excess dripped off through the filter and into the aquatic chamber. The carbon cycle occurred when plants or worms died in the terrestrial biome, putting carbon into the soil and air. Then the plants absorb the carbon through their roots, and the excess dripped down to the aquatic chamber. The nitrogen cycle occurred when nitrogen was soaked into the soil from the worms defecating and the plants absorbing it. The excess nitrogen washed down to the aquatic chamber, and then the water from the aquatic chamber was poured back into the terrestrial chamber. The phosphorus cycle occurred when plants or decomposers absorbed phosphorus from decaying plants. The excess phosphorus drips down into the aquatic chamber and is then absorbed by the aquatic plants. Question 3 asks, is your ecosystem column a closed or open system? Our column was an open system since we poured the water in ourselves and it was not self-sustaining. Question 4 asks, discuss 5 limiting factors in your habitats. 5 limiting factors would be our size of the ecocolumn, since it was about the size of a large water bottle. The amount of sand in our filter, which didn't allow nutrients or some types of nutrients to drip down to the aquatic chamber. The amount of water in the aquatic biome was also a limiting factor since it was such a closed area. And the amount of dirt in the terrestrial biome, since it's also about the same size as a small water or a large water bottle, and the amount of sunlight the plants could receive since they were only receiving it from a lamp.
This is the analysis questions, 5 through 8. Number 5 says, explain what eutrophication refers to and how this occurs. Apply this explanation to your ecosystem column. How might eutrophication take place in your column, and to explain fully. I said that eutrophication is when the runoff from other sources are high nutrients because of man-made products. In most cases, it causes an algae bloom within the body of water. In turn, the algae bloom causes the water to lack many key items for the survival of the ecosystem. Oxygen for the fish and other animals becomes scarce because the algae uses and blocks all of the oxygen from getting to the deeper water, and blocking of the sunlight causes nothing to grow under the algae, causes the whole entire ecosystem to collapse. Eutrophication could only happen in our ecocolumn if we added too much fertilizer, which for us was leaves. If we added too many leaves, the leaves themselves would have overpowered our plant and would not allow the proper light to reach our plant and its roots. In addition, the leaves would take a long time to fully break down and become a viable source of nutrients for our plant when there are too many. Number 6 asks, leaching is a process by which solid materials such as minerals are dissolved in a liquid, which is usually water, and transported to another location. Explain how leaching could occur in our ecocolumn and what possible impacts of leaching to organisms in our ecocolumn. I said that leaching could only occur in our ecocolumn if we overwatered our plant. The overwatering of our plant mixed with the perfect amount of fertilizer for the proper amount of water would be destroyed because the overwatering would cause the minerals and nutrients to all leach out too fast for the plants to absorb any. Then our soil would no longer be holding enough nutrients for our plant, and the plant would begin to die, and if we didn't replace the lost nutrients, the plant would completely die. Question number 7 asks, what evidence of ecological succession was taking place in your column? And if you did not see any ecological succession, explain what it would have looked like. I said in our eco column there was no evidence of ecological succession, and if there was any ecological succession, it would have probably presented itself as our plants dying in the beginning. Then the other plants would begin to start rebuilding the ecosystem and would return to full health once again. Question number 8 asks, what type of biome does your ecocolumn most closely resemble? Defend your choice. The biome that most closely resembles our ecocolumn is a rainforest because our ecocolumn took in and used a lot of water on a daily basis. The amount of water we put in resembled the high levels of precipitation in a rainforest environment. The shade caused by the leaves helped to keep the worms below alive as the soil temperature stayed low as a result of the plants blocking the light. In our ecocolumn, Our ecocolumn could never be a tundra because the temperatures are much higher than that of a tundra, and it could also not be a desert because the levels of water are much higher.
Okay. Our goal in the experiment was to examine the different food chains and webs and look at the interactions among the different chambers. It was also to identify and see how the abiotic and biotic factors affected each other. Our first chamber was on top, and we started with seeds and dirt and then also added worms. We watered the plants, and after the first week, they began to grow. The chamber below that was used to filter the water did so through sand and gravel, with the finer particles being at the top and becoming bigger as it funneled downwards. The third and final chamber was the aquatic chamber, where the filtered water deposited and supplied nutrients to our aquatic plant and fish. The results the test yielded were a surprise, as we did not expect the plastic bottles filled with dirt and plants to create a sustainable ecosystem with the chambers of sand and water. We found that the worms aerated the soil enough to foster plant growth, and that the plants that grew thrived in the environment. We also found our betta fish left bubbles in the water of the aquatic chamber, which we researched and found that it was because it was happy in its environment. There was a possibility of error in the measurements we took, but the results were not skewed enough to show any real errors. I assume that there could have been error when using instruments to record data. An improvement to the lab could have been repeating measurements to ensure the validity. Another improvement could have been taking measurements twice a week to see if there was a change in the ecosystem between our measurements. The ecocolumn can be related to life as it needs balance to thrive. In the system, the plants, worms, and decomposing material are all things that need to be precise. In life, maintaining success requires balances between many things like play and work, and even in relationships, church and state. It creates a balance. Say, watering our plants on a daily basis is like doing homework. If you don't do your homework daily, you'll get behind in just a day's time. Same goes for watering our plants, except for the consequences are much worse because we cannot just go back and water our plant. One ecological problem that we deal with on a global scale is water pollution. When water seeps through the ground, it is filtered and enters aquifers, which many people rely to live off of. This process can be seen in the first two chambers of our ecocolumn, where the water is filtered through the dirt, sand, and gravel. Fertilizers around the world contaminate this water, which leaves the water undrinkable. We did not completely clean our water in chambers, but we can tell that it was clean by the time it reached the third chamber by looking at water pH levels. Our research can tell us that if the water were to go through more filtration, it would be safer to drink. A second ecological problem is food loss. Around the world, 40 million tons of food is wasted every year. Our research found that when wasted food, such as leaves, is left to decompose, it raises the soil fertility levels. We should put more food back into the soil to help our plants grow. And one last ecological problem the world faces is the loss of biodiversity. We saw this in our lab as some plants grew at the expense of others in our ecosystem, and it lost biodiversity. This happens all around, and the takeaway is that we need to have enough space and nutrients to let each species thrive. European Parliament News reinforces our claims that the environment needs sufficient space in order to stop the decline of biodiversity. NRDC.org also matches our claim that water needs to be filtered well in order to be used by humans, and that fertilizers and pesticides are a leading cause of water pollution. Cedar Circle Farm Education has information on composting food and waste into fertilizer, much like how we use plant waste, the leaves, to fertilize our ecocolumn. After completing this lab, we should take our analysis and make it useful. In the future, if we plant a garden, we should check the soil fertility and get worms to make sure the ground is suitable for growth. A question that I would like to answer is, how is nature balanced enough to sustain itself, if that makes sense? Another is how such a system comes to be on a rock floating through space. And one last question is, do these processes happen on other planets with life, or do they have different cycles and processes that sustain life? Over the course of our experiment, we saw how decomposition leads to the growth of plants and how abiotic factors like dirt and gravel do the important job of filtering water so that marine life can thrive. We found that closed ecosystems are not kind to biodiversity, and only those suited to the environment will thrive. We also found that soil quality really matters and saw how cycles such as the nitrogen and carbon cycle control the environment.